I think the Comprehensive Cancer Centre that we have here gives us the ability to really try to tackle cancer through the entire experience that a patient might have and work out how we can improve outcomes every step along the way. I really join this battle at the stage of when tissue is made available from a patient's tumour. And I'm involved in two particular studies. One is to study the DNA of that tumour, to understand why that tumour developed, try to understand why it's growing aggressively or not so aggressively, trying to see if we can personalise a drug therapy towards that particular tumour. And that's all based on the DNA. The second approach that we have is that we can take those same cells from the patient and we can start to grow those in the lab, but do it in such a way that we can systematically screen drugs almost real time to try to come up with a readout where we think there may be the optimal drug therapy for that patient. These are in the research stages now, but in this sort of place we can really accelerate that towards clinical trials. In the very near future, our goals are to, as close to real time as possible, be able to get the full genetic readout of a patient's tumour, select the drugs that we think are likely to work best on that tumour, and also have a matched organoid culture system where we're growing those cells real time and we can test those drugs before taking that onto the oncologist and his team deciding how they would use that information on the treatment of that patient.